Okay, it's 3:30, so I will start. Thank you everyone for coming to my talk. This is part of the Nebraska Water Science Center's seminar series. As I mentioned, Ann, who usually runs this, she's not here. So I'll introduce myself and say I am an associate professor in the School of Natural Resources right here in this building. And I have been here for a number of years. And I built my talk around a bunch of the work, though not all of it, that we've been doing in my lab, really trying to highlight work done by my grad students and undergrads. And in the picture in the title slide, you'll see Kayla and Matthew, who are two undergrads that worked with me for the UCARE and Cabela's support. And what you might not be able to see is that we're looking at a really small petri dish, and in that petri dish contains a bunch of sand. And sand makes up a lot of the bottom of the rivers and streams in Nebraska. And it makes for an interesting habitat to study because most of what we know about our streams and rivers comes from those that have been studied with hard surfaces. So that'll come up maybe a little bit later again. But anyway, I would like to also thank the many other students and staff and postdocs that I've had the pleasure to work with since I've been here, and my collaborators, which I will not read because I do not have that good of eyesight to read that computer screen, and I don't want to forget anyone's names, and you can read them all. And of course, thanks to all of the state, federal, and local agencies that have supported this work in a variety of different ways. So, it's probably pretty safe to assume that everyone thinks water is important. And this is a quote from President Bill Clinton a number of years ago, but I think it really highlights well that it's probably a unifying theme that we all think that we want healthy drinking water, that we would like kids to be able to go swimming in lakes and rivers and not worry about their safety. And, you know, maybe that we want to go fish in those waters and perhaps eat the fish that live in those waters. And probably everyone in this room feels that way as well. And here's a picture of a creek that I was just at a couple weeks ago that really exemplifies those characteristics, except that it doesn't have fish. It's a perennial stream, so not for any bad reason, just because it's a perennial system. So, yeah, and this is also another old quote that I found from a Lincoln Water Supply publication in, I think it's the 1930s. But it also just sort of says, you know, even 100 years ago, this was an important thing and something that the university and those of us in the university have been really striving to do for the last 100 years, right? Let's manage the natural resources of our state. Unfortunately, right, there are difficulties in doing that. Lakes close in Nebraska routinely in the summertime due to harmful cyanobacterial blooms. You might know those as harmful algal blooms or HABs or cyanobacterial blooms. And they can become problematic because those cyanobacteria, some of them can form compounds known as cyanotoxins that if ingested or rub on your skin can be an irritant or a hepatotoxin, a neurotoxin, and cause a lot of issues. They're also really, really hard to remove from drinking water. Toledo in Ohio gets its drinking water from Lake Erie, and Lake Erie has been struggling with a lot of these blooms, and you know, there was a time that they had to actually shut off their drinking water source and rely on bottled water. And this is just such an incredibly unfortunate thing, right? The fact that we can generally turn on our faucet and feel safe about drinking the water is a really basic human right. And these cyanotoxins will threaten that. This is also where, if you've heard my talk, give a talk at any point, you know that I like to make a public service announcement, which is if you have a pet and your pet likes to swim in the lakes around here, do a really good job of washing your pet off afterwards. Dogs can get a uniquely high exposure to these cyanotoxins because when they get out of the water, they lick their fur. So if there are cyanotoxins, whether it's in the cyanobacteria or just in the water, they can get a really high concentrated dosage. So please wash your dog off after they swim. People too, although we don't tend to lick ourselves clean. So, okay. Other wildlife, right, also can be at threat for these pollution levels. There was a really kind of tragic story of these pelicans. About 90 pelicans died from a reservoir in Greece. I think it was related to cyanotoxins because they found such high cyanotoxins in the levels of those birds. It's a concern for our cows, livestock, other fish as well. And fish also need oxygen in the water to breathe. And so these blooms can deplete over time the oxygen in the water too. So that's sort of one big thing that is a problem that we have to think about in our waters. And then the other one, right, is nitrate. Nitrates in our groundwaters. This is a US map. Nebraska is right there in the middle, bright red, because it has pretty high vulnerability to nitrates in drinking waters. And you live in this state, you've seen it in the news, you know this is a concern. But the concern, of course, is that if you are drinking water with high nitrates, that nitrate can swap out with the oxygen in your blood. A particular problem to young and old. And this is a headline from a case of a baby in Wisconsin that died of blue baby syndrome. We've known about blue baby syndrome. We have really strong regulations on nitrates in our waters. And so the fact that there are still people that are suffering from it is quite a shame. I will say, too, that those strong regulations are for municipal water sources or wells that serve over 250 people. So if you're on a single family well or smaller, you don't necessarily have to comply with those regulations. And so you can be uniquely vulnerable to nitrate levels. Okay, so the cost of nutrient pollution in waters. I have here a couple pictures of what is probably my favorite case study of a city taking good control of their natural resources, right? New York City. New York City gets its drinking water from reservoirs in the Catskills. So they have a beautifully clean source of drinking water for one of the largest cities in the world, right? It is no hard thing to say that this is part of the reason that New York City was able to thrive as much as it was, due to clean drinking water. Water treatment costs. There's a recent study looking at water treatment costs and found that about a third of large cities have had to spend more than $5 billion a year, right, due to watershed degradation that causes issues like I just sort of went over. In the US, we have tried to support our watersheds, and we have spent that to the tune of $5 trillion since 1972 to meet water quality standards. So we are putting money in, which is great, and it would cost us even more if we weren't. All right, so Nebraska's nitrate problem, we have one. It is in our groundwaters, and it has recently been in the news a lot. And again, here's sort of another case study of why it matters. So Hastings, Nebraska needs to get rid of the nitrate in their water to a tune of $15 million. 25,000 people live in Hastings, which means that every person needs to basically pay an extra $600 a year for the clean water to get those nitrates out, because it is really hard to get nitrates out when they're in, although not as hard as the cyanotoxins. So what does this mean? You could just alternatively buy some drinking water, right, bottled water. The average gallon of bottled water is about $9. I looked it up a couple of minutes ago, which means that if you drank about a gallon a day or used about a gallon a day, that would cost you about $3,000 a year. So $600 is certainly cheaper than $3,000, give or take, depending on your water usage. But yeah, so certainly worth supporting our drinking water treatment facilities. Okay, so if I could sum all of those last slides up into two pictures, it is that We like to have our nitrogen and phosphorus in our fields so that we can grow our food, but we do not like them in our waters. We don't want the nitrate there, we don't want the phosphorus there, because it causes growth of things we don't want, the algae and cyanobacteria. So where are these imbalances particularly striking? There's another really recent report out of some scientists at the EPA looking at agricultural surpluses. So this is basically when, based on their calculations, too many nutrients are going into these watersheds and are leading to where those nutrients are going to go, generally to lakes, rivers, streams, wetlands. So you can see it's a pretty broad view, but certainly for both nitrogen and phosphorus, Nebraska is in a surplus. Although I always have some issues with these broad maps, right? Because a lot of interesting things happen locally. And yeah, anyway, but broad view, right? A lot of extra nitrogen and phosphorus is still going on our landscapes. This is a zoom in of phosphorus sources because phosphorus can come from other sources as well. So things like human waste, so that is our treated or untreated wastewater. Phosphorus isn't always removed in that process. A little bit by atmospheric deposition, which is largely dust. Fertilizers and livestock waste. And so in Nebraska, perhaps not surprisingly, farm fertilizers and livestock waste are a big contributor to phosphorus. Okay, so again, right? Great to have our nitrogen and phosphorus in our field. We don't want it in our waters. There are lots of recommendations out there to think about conservation. In Nebraska, those strategies tend to relate to reducing how much stuff we put on the field, restoring wetlands, and both in field and around the field, and creating non-wetland buffers. So slowing the nutrients down in the landscape. This is all great. This is not work that I do. This is work that a lot of other people do here at UNL, and that's wonderful. What I like to think about is, okay, so what happens when those nutrients get in our lakes, rivers, and streams, right? Our lakes, rivers, and streams are not just passive pipes. They don't just take those nutrients and move them to the Gulf of Mexico. They process them. Globally speaking, that number is around 50% or more. So of all of the nitrogen and phosphorus that gets in a lake, river, or stream, about 50% of it either stays in that ecosystem, or for the case of nitrogen, is denitrified and goes back to the atmosphere. That's pretty incredible, right? We're burying a lot of nutrients in the sediments, and we're denitrifying a lot of the nitrogen into the atmosphere. The caveat, of course, is while these numbers are a good starting point, they rely on mostly studies that come from temperate systems, not necessarily from grasslands or deserts or tropics. And so there's a lot we still can learn about what these aquatic ecosystems are doing. And, you know, here in Nebraska, what our streams are doing. Are they reaching that 50%? Are they way far from it? Things like that. And so that really has been the thrust of my research, right? Looking at how Nebraska aquatic ecosystems are responding to inputs of nitrogen and phosphorus, and you can read that. I can't, but yeah, that's fine. Okay, anyway, so if we know how the nutrients are cycling in our aquatic ecosystems, I would say we can better manage those aquatic resources. To start at this question, I worked with my first master's student and lab technician, Alexa and Murray, to look at nitrogen and phosphorus cycling in Nebraska's small streams. Generally speaking, small streams tend to do a pretty good job of denitrifying nitrogen and possibly taking up phosphorus. So I sent the two of them out to a bunch of streams across the state to look at nitrogen and phosphorus uptake in these streams. We do this by a process called nutrient spiraling, What that is basically filling up a carboy, that bottle there in the picture, of a concentrated amount of nitrogen and phosphorus and a conservative tracer, dripping it into a stream at a known rate, and seeing where those nutrients and tracer go downstream. So we add the nitrogen or phosphorus in, and it spirals along. How quickly that nitrogen and phosphorus is taken up is a proxy for what we call an uptake length. So the shorter that uptake length, or the shorter amount of time the nitrogen or phosphorus moves in the stream before an algal cell picks it up or bacteria cell picks it up, the shorter that uptake length will be. So we can think of that sort of as a proxy for reactivity. The more reactive the stream, the more potential for those streams to take up those nutrients, to make that 50% higher. There's been a lot of these studies across the US and the globe. It's a pretty common metric that we use. And kind of regardless of stream size, the reactivity doesn't change a whole lot, which means that our smaller streams, right, tend to be pretty reactive. They sort of punch above their weight even though they're smaller. So we wanted to see where Nebraska streams fit in this collection of work that's already been done. And we found out that the uptake length in our streams are pretty long. So they're not very reactive, which suggests what is going on. And some more work that sort of came out around the time really started to critique these methods, that they don't really work well in conditions where nutrients are high. So Alexa, in addition to doing all of those studies, she also just looked at the concentrations of phosphorus as SRP and nitrate in the streams and found that, they were sort of on a higher level. I'm not going to tell you what that means in these graphs, but this is all just to say that the nutrient concentrations are high. So maybe this technique of looking at uptake isn't so great. So now we are going to go to Scotts Bluff, where we're trying out some different methods to get at the same question of nitrogen and phosphorus uptake potential in these streams. And throughout this talk, we're going to kind of jump across the state, and so this map is going to be sort of a roadmap for the rest of the talk. Okay, so the streamnet project is funded generously by the Nebraska Environmental Trust and supported by the North Platte Nebraska Research District, and done in collaboration with Steve Thomas while he was still here at UNL, and Chris Chizinski, who did some social science work on it that I'm not going to talk about here, but happy to discuss more. Okay, so the idea of streamnet is that there are these new machines that do automated testing for nitrate, phosphate, and ammonia. That means that I don't have to send myself or my students out to a stream to collect water, filter it, bring it back to the lab to analyze. That all happens in this box. Steve and I are very excited about this because of the potential to reduce human labor involved with water shortages or water collections. We did not design it. It was designed by Vince Kelly out at Green Eyes in Maryland. But the other reason we are so happy is because we got this funded in 2020. We put our order in right away, and the box didn't come until like the middle of 2021 because of delays with COVID and supply chains, and then the box got lost in freight for about 3 months. It was an ordeal. Anyway, we were quite happy when it got set up, and we also had plans to have an automatic data portal so that anyone could see the data right away. So we set up the instruments our first year in 2021. We were able to look at oxygen, temperature, conductivity, the nutrients that I mentioned. That's one of our field sites. And 2021 was really just a year to figure out if these instruments were working, how to get them reliably producing data. In 2022, we set them out again. We got some data, and then one of our units was stolen, and flood came and got rid of some other parts. So our 2021 or 2022 data is pretty slim, but by 2023, this last summer, we really got our act together. We had 2 instruments out there, more or less successfully getting data for the ice-free season. And so here is a picture of that, which you can't see. I don't know. Okay. Oh, that's really sad. You can't see either graph. Okay, so what these charts would show you is a bunch of data that the nitrate I pointed out, those circled areas are where nitrate levels are around 10 milligrams of nitrogen per liter, which is at that level that is of concern for drinking water. So the instruments, because of their much more high frequency sampling, are able to get at these shorter term events when nitrate levels are too high, which is pretty exciting. The other exciting thing is that what you can't see but you can imagine are beautiful sinusoidal curves on this 2 weeks of data. And you would see 14 curves representing the increase and decrease of these nutrients each day. So showing the fact that there is biogeochemical activity happening here, organisms are taking up those nutrients, meaning that despite the fact that we couldn't detect those uptake levels in Alexa's work, we can using these instruments. So we're pretty excited about that. We're gearing up for 2024 when we'll have both of those units back up and running. We have 2 more sort of in the wings once we get our personnel working to help get a broader view of nutrient activities in the Scottsbluff area. So apologize again that those data are not showing. Okay, so in summary, right, we might be able to do a little bit better job of detecting biogeochemical activity when we are using some new techniques. So more to come on that. My dream for StreamNet is that we could get these instruments across the state. If you have an extra 50K lying around, come talk to me and you can be a sponsor of one of those units. All right, so next we're going to move up to the Niobrara River. And just by a show of hands in the room, how many people have been up to the Niobrara before? Okay, that's great. You know what a beautiful place the Niobrara is. It is a designated National Scenic River, mostly fed by groundwater. The Q here on the bottom of this graph is our discharge. So it has pretty steady discharge year-round, and although it does show a little bit of the irrigation drawdown in the watershed, but really beautiful river, and it's makes a nice contrast to some of the other systems that we've been working in because it tends to have lower nutrients in this aquatic system, and so it kind of could be a potential more control of how our rivers could function in the state. This work I began in 2019, and if you can remember back to 2019, we had quite the event. Right, the bomb cyclone came in March 2019, flooded much of the state. This is a picture that I took. I was flying out of Omaha Airport shortly after we got those rains on frozen ground, and you can see that road, that farm right out of Omaha is all underwater. And the same thing happened well, not quite the same thing, but the Niobrara flooded as well. Here's another version of that discharge graph. So discharge being the amount of water that's moving in the system. All of the dots represent the daily values for that month. This is all the pre-2019 data. This is 2019. You can see it started out as a pretty normal year, but then March 2019 happened. Discharge went up to, I think the highest recording was about 300,000 liters per second. And it stayed elevated for much of that year, continued to stay elevated in 2020, and wasn't really until 2021 that those levels returned back to normal. At that time, Matthew Chen was working in my lab, and he knew that rivers are really important for transporting nutrients across the landscape, and so he wondered, what did this flood do? He went into this question thinking, well, we know phosphorus likes to stick on to particles, so phosphorus concentrations will probably track discharge, become elevated when we have the flood, and then return to normal. Nitrogen, we really weren't sure what was going to happen. We suspected dilution because nitrate's in our groundwater, so if you get more surface water, that should dilute out the nitrogen coming from the groundwater, but we weren't sure. But as I mentioned, we didn't start working in the river until 2019, so we didn't have any pre-flood data, but fortunately, a bunch of the National Fish and Wildlife Service and the National Park Service had been collecting water quality data from the river beforehand, so he was able to harvest that data, add it to our own collections, and then write up a really nice paper looking at how the loads and concentrations of nitrogen and phosphorus responded to that flooding event. And so between before and after the flood, you can see a really strong decrease in nitrate and a strong increase in phosphorus, which kind of made sense. What made a little less sense was the fact that nitrate levels persisted to be low after the flood. There's no reason to think that they shouldn't have rebounded, much like the phosphorus did. We collected data again the following year, and it really took until those 2021 samplings until the nitrate levels returned back to normal. I don't know why this happened this way. We tried to look into some of the groundwater data to see if there were changes there. I haven't really found a good data source yet for that. So if you know of any, please come talk to me. But this would be a really good thing to figure out, right? Like, why all of a sudden do these nitrate concentrations decrease, and why did they stay low for so long? Is it somehow that the flood ramped up denitrification, or did a bunch of nitrogen get flushed out of the system? Was there some change in watershed use? I don't know. So it would be really good to figure out, because that could help us manage nitrate concentrations moving forward. So the other thing that came out of this, right, is this legacy of extreme floods. It wasn't just that first year after the flood that we saw the effect on the ecosystem, but it was still 2 years after until the ecosystem really rebounded from that flood condition. So when we're studying these extreme events, long-term data are important. All right, so from the Niobrara, let's go to the Elkhorn, and actually we won't do much there, but I just wanted to give a shout out to some new work that I have funded with Paul Ayaye and David Manning at the University of Nebraska, Omaha, where we're diving into these questions about the river ecology in Nebraska, or in the Niobrara and the Elkhorn River. I'm not talking about the data today, but I do have longer term data on periphyton abundance in the Niobrara, nutrient limitation of that periphyton, and we're doing the same collections in the Elkhorn. Paul is also looking at the microbial communities in those systems, and David is also doing some other ecosystem metrics, looking at metabolism and primary production and respiration in those systems too. Okay, now let's move to lakes, and that is the Sandhills region out in western Nebraska. And the question here is that if a lake has a lot of nutrients, does it matter if it gets more, right? If we've already saturated our aquatic ecosystems with nitrogen and phosphorus, does it really matter if we add any more, right? Are we going to see a difference in growth in those ecosystems? And this is an important question for Nebraska, because if we look across our state, about 60% of our lakes could be considered eutrophic. They're already saturated with nitrogen and phosphorus. So if we think about those blooms and cyanobacterial production, do they respond to more nutrients or not? And this is some work that my PhD student, Daniel Geschwenter, is working on. He's doing a lot more cool stuff. I am just picking out a little bit of it for this talk. But he took advantage of the fact that the lakes in the Sandhills region, you do see a span of concentration of nutrients in those systems. Some of them have really high values, some of them have lower values, as well as some of them are more along the freshwater continuum, and some of them have a lot more salinity in them. These gradients are largely related to how connected the lakes are to the groundwater in the area. So if they're less connected, they tend to be more evaporitic and concentrate stuff in them, versus they can get, if they're more connected to the groundwater, they tend to be fresher. It's a simplification of it, but that's where we are. What I think is really kind of amazing in these lakes is that you have Border Lake, which has over 1 milligram per liter of phosphorus in it, which is just, I mean, it's incredible. So before I came to Nebraska, I was working in lakes up in Wisconsin, where there's another, I mean, the whole state is basically a lake district. So you had another lake district kind of like this, a bunch of lakes together in the landscape, and the lakes up there, like if they were over 0.1 milligrams or 100 micrograms per liter of phosphorus, people would freak out. Usually it was like 0.01. So magnitudes of order less phosphorus in those lakes up in the forested landscapes of Wisconsin, versus down here, yeah, just really incredibly high numbers. So these are the systems that you think would not respond to nutrients, right? They've got all that they need. Well, it turns out, could be a different story. This is before I jump into the results, here's a close-up of those lakes. So if you just take a plastic jar, put some water in it, that's what they look like. And you can see there is quite a bit of growth in Bean and Border Lakes. All right, so Daniel went out there repeatedly in May, June, July, August, took jars of water, diluted them so things had some room to grow, added either nitrogen, nitrogen and phosphorus together, or neither, and looked at the growth response. Also added trace elements, which is TE. So those are the micronutrients that might be important cofactors in different enzyme activities. So sort of a like really squinting your eyes, what's going on here. In our most nutrient-rich lake, they're still responding to inputs of nitrogen and phosphorus together, right? We can still get a little bit more growth. In the other high or medium nutrient lake, we see some indication of nitrogen-phosphorus co-limitation, nitrogen limitation in Island Lake, and then phosphorus limitation in Smith. Again, I am super squinting my eyes to making these assessments. Daniel's got way more information about the nuance of the different responses. But this is all to say that even though these lakes are really eutrophic, they could grow even more things in them. So when we think about managing lakes in Nebraska, yes, things will respond to more nutrients. They can get greener. And you also need to think about nitrogen and phosphorus, not just one. Some of you in this room may have been trained up under the phosphorus paradigm, that phosphorus is the only limiting nutrient to lakes. Let me tell you that that is not true. It can be, but it can also be nitrogen. The flip side is you might think that, we live in Nebraska, we have to think about nitrates, and you're therefore ignoring phosphorus, and I am here to tell you, you can't ignore phosphorus. You have to think of phosphorus too. So I wanted to dive in a little bit more with nitrogen and phosphorus and go through just a little bit of a thought exercise. Microcystins, or microcystin is a type of cyanotoxin created by that organism, Microcystis, there on the slide. It's a form of cyanobacteria. And this work from Ortahel et al., surveying a bunch of lakes in Canada, found that you tend to get the most cyanotoxins at low nitrogen to phosphorus ratios. So low nitrogen to phosphorus ratios means a lot of extra phosphorus compared to nitrogen in that ecosystem. That tends to be watershed-wise related to areas where you have CAFOs or lots of animal production. So taking that number, here's another look at total nitrogen versus total phosphorus in the system, and where you get those colors of the dots indicating more cyanotoxins, it tends to be when you're below this threshold of about 23. So to the extent that we can extrapolate things from Canada to the US, to our lake systems, I was curious to say, okay, we've got a lot of data on nutrient levels in our lakes. How many of those lakes fall below that N to P ratio? And it turns out, not an insignificant amount, right? So this is from a survey from the EPA from 2017, and it certainly suggests that a number of our lakes could fall below that threshold, meaning that they might be lakes more susceptible to those cyanotoxins. We also have good data on cyanotoxins, and so sort of the next step of this project is to start looking at those connections a bit more. But I just want to say it again. Are we doing enough in Nebraska to manage phosphorus? No. The answer is no. Okay. Moving now a little bit more locally. Here we are in Lincoln, and I don't quite have all the time to go through the other projects that are currently happening in the lab, but I wanted to bring some up. So I have another PhD student in the lab, Uche, who's really interested in the reservoirs around Lancaster County. He's looking at inputs into the reservoirs and outputs of those nutrients, trying to figure out how water level in the reservoirs relates to nutrient retention capacity. It's going to be a pretty exciting project, but it's just started up, so I don't have a lot of data there. And Jace Miller's a brand new PhD student in my lab, also interested in phosphorus dynamics and the role of viruses in moving phosphorus in our aquatic ecosystems, and whether or not we really understand phosphorus cycling in those systems. And this is a good point for me to bring up. Yeah, our aquatic globally, sort of that like 50% number, we're concentrating phosphorus in our sediments. That doesn't mean that that phosphorus stays in our sediments in the lakes. It can be internally, we call it internal cycling, but it can get back up into the water column where it can promote further algae and cyanobacterial growth. So Jace is looking to see what viruses have to do with that process. And finally, I wanted to bring up Josh Matthews' work. Josh Matthews is not a Daphnia, but he is studying them. That is our charismatic microfauna of lake ecosystems. It is a zooplankton, which means it's a little animal that floats in the water. It tends to eat algae and cyanobacteria. So a question is, how are the zooplankton in reservoirs around Lincoln responding to all of this growth of algae and cyanobacteria in the lakes? How much do they eat? Because if you have a lot of zooplankton in your lakes, that can really help reduce the burden of algae and cyanobacteria in those systems. But they have to eat them. So that is what Josh is looking at. So just to sort of sum up, we're really excited about some of the new technology that we're using in our lab that might give a better sense of these eutrophic prairie grassland systems, what's happening with nutrients there. The other one, right, is that eutrophic lakes can still respond to nutrient additions in ways that we are still beginning to understand. And some of the opportunities that I think exist out there for better natural resource management, understanding prairie streams a bit more better and what they are doing with sequestering nutrients means that we need to use new techniques to really get at what's happening. And then finally, what is that missing nitrate? Where did it go in the Niobrara River? I'm really excited to answer that question moving forward. And at the end, I will put a picture up of a stream here in Nebraska that is clear water. We don't see many of them, but they do exist. And you might know where this is, and I might answer if you ask. So thank you all for your attention, and happy to take questions if there are any.
So with your instrument that you had in Scottsbluff, you were seeing those peaks, recurrent peaks of really high nitrate concentrations. So what's pulsing that in the system that you were missing before?
I think, so for the new labs, they're collecting a nutrient sample like every hour, or it's actually every like 2 or 3 hours, but we're getting multiple data points in a year. When we do the nutrient injections, the whole entire experiment takes about an hour or two. So you're not going to see that level of change that you will in that full 24-hour section. So it's a much easier way. Well, it's not easier, but you get a better picture of what's happening throughout the whole day. If Alexa had sat streamside by any of the streams for 24 hours and taken samples, she probably would see that diurnal cycle as well. But I guess she needed to sleep. It's the diurnal thing. Yeah, and so, I mean, and that's the interesting part too, right? Because if it's diurnal, but it's always the same baseline, it's not a net uptake or loss of nutrients in the system. Work I didn't show here, but that was done by my master's student, Annie. She took advantage of the fact that in Scotts Bluff, the water that is used in the field is kind of taken upstream on the Platte. And then it goes through this you have these canals flanking the river for a bit, and then they rain down into the river. And so she was able to get, by some strategic spatial sampling and some mass balance equations, she was able to look at the flux of nitrogen and phosphorus off of the landscape or out of the river through those, and she found some really interesting monthly patterns there. So we're kind of we're excited to sort of couple this really fine-tune daily cycle stuff with sort of these bigger spatial pictures to get a better understanding of what those systems are doing. Yep.
There's a question online about Verdigris Creek.
Oh, it is not Verdigris Creek if the answer is where this stream is.
Oh, okay, yeah, that's, I think, what the...
It's Long Pine.
Ah, okay.
I haven't been to Verdigris, but now I clearly need to go because it's a clear water stream.
Okay. Any other questions?
I think.
Oh, sorry.
Considering that so many of the streams around here are actually canals that flush through really high amounts of water, depending on downstream need, how much potential is there actually for biogeochemical processing? I mean, if you have these really intermittent high discharges, there's not going to be a lot of algal growth there to take up nutrients. And I'm wondering if any of Arnie's work or any of Riley's work have actually been able to look at that.
Yeah, so in Alexa's work, the two streams that she sampled that we actually were able to detect uptake were the canal systems here in Lincoln. So even though they're completely channelized, they just get such high sun and high nutrients that you can grow a bunch of algae in there that take up those nutrients. The flip side, right, exactly to your point, Daniel, is that when you get a giant flushing event, depending on if that nitrogen was denitrified or stayed in the system, it's just going to get flushed downstream. And the phosphorus is just going to get flushed downstream for sure. But depending on where it goes, does it go into a reservoir where then it sinks out? We know reservoirs are really good at capturing nutrients, for better or for worse. They fill up pretty fast around here. Yeah. In terms of Riley's and Alexa's work, or Riley's and Ani's work out in Scotts Bluff, there is always this sort of when you get these really nice daily cycles, how does it scale up? You really do need to understand the flood events to get the bigger picture of where the nutrients are going. Yep. Which I'm basically just agreeing with what you said.
I'll ask a question if it's okay. The equipment that you were putting in the field, for how do you see that as a trend, and what would be the optimum number of that kind of thing on the landscape in Nebraska?
Yeah, that's a great question. So they probably need about a 0.1 to 0.2 FTE per area to maintain. So even though they are automatic, they need, someone needs to go out there and check them every week or two to refill reagents, just make sure things are working well, that type of thing. And we have this great relationship with North Platte NRD, and so their technicians, Keeley for sure, is doing a lot of great work keeping those instruments up and running. In Scotts Bluff, our original plan, and we originally had 3 units up. One was from a watershed that represented more of an urban influence. One represented more of the row crop influence, and one was row crop and CAFO. So from a nutrient management perspective in a watershed or in like a natural resource district, having representation from your dominant landforms can go a real long way into getting a good picture of those internal daily cycles, and then scaling up to a year, where are the nutrients really coming into the system, when are they getting flushed out, when are they staying, when are they getting processing. The USGS does an amazing job with water level gauging, and the technology is a lot easier than these types of things, but sort of mapping onto that general framework would also be a really smart way to extend the data that those systems are generating.
Are there other states that have those kind of systems or networks in place?
Yeah, the biggest network is on the Maumee River. So Laura Johnson, who gave a talk here a few years ago, she's got like 20, 30, 40 across the state of Ohio, and they're getting really great information about where the phosphorus is coming off the landscape because of Lake Erie suffering so much from these cyanobacterial blooms. And one of the key pieces of information that she's been able to generate using these systems is the role of phosphate leaching off, and it likely being related to oversaturation of fields with manure. Using manure to fertilize fields can be really, really great, but what tends to happen is it doesn't go far enough, and it tends to get locally applied. And we've seen this in Ohio. We're starting to see it in southern Wisconsin, where you're just getting these really phosphorus-saturated soils, and suddenly there's phosphate in the groundwater. And what's happening there? There's phosphate in the groundwater in Nebraska too, which is one of my many things on my list I'm interested in diving more into. But yeah, so in short, yep, folks are using these in other places, and I think there's a lot of potential here in Nebraska too.
Very good. Great. Any closing questions? I'll look real quick online. No? No one? Actually, that was a very good segue. Thank you very much for the speaker next week, who's about livestock, livestock waste management. So very good. Well, let's thank one more time.